All right, and thank you all for coming to the Biome webinar series. So for those of you who are just joining, I have a link in the chat. I'll send it again because if you want to hear more from us, if you want to hear about these webinars, if you want to hear about the speaker series that our iGEM team is running, then yeah, the email list is the right place to be. So today we're going to be going over this concept called chromatography, and I was looking online trying to find some analogy, to compare it to, but I couldn't really find a good one. So I came up with my own, the tortoise and the hare, but it's the bio edition, not Aesop edition. It's bio edition. Now, before I get into sort of the technical scientific aspects, let's first look at this word, right? Chromatography or chromatography. Chrome, it's color, right? So colorful things typically have the first prefix as chrome. Like chromium is a colorful element. And graphy is writing. So chromatography is really, they first discovered it by separating colors out from leaf pigments. They were like, oh, look, you can have these colors that can essentially migrate at different rates. And that then developed into a whole bunch of other techniques that can then allow scientists to now separate things. A big part about science, and especially biology and wet lab work, is about I have this mixture of stuff. I have this cell, juice inside of my flask. How do I get exactly what I want? How do I get the protein of interest to study or to do reactions with, for example? And so, yeah, that's the basics of chromatography. It's separating things, right? And the rest of today's talk is going to go over the word how, right? How do we separate things? And what techniques should we use and what properties should we take advantage of to separate these things? Now, I'm sure you've all heard about, the story about the tortoise and the hare, right? I'm not sure about the the specific story, but this is like my mental version of it. I haven't read it in a long time, but at least in my mind, I'm just like, oh, well, there was this tortoise and the hare, and they were like, oh, well, I'm faster than you, and then they started racing, and then the hare was just super fast. He was just booking it, and the hare runs super duper duper fast, but then he takes a nap in the middle and he like finds some carrots or whatever. I don't know. He slows down and because he's lazy and he thinks he's really good, right? And he thinks he's gonna win. So the hare takes a nap. The tortoise slowly, passes the hare and then wins in the finish line. So that's where the phrase "slow and steady wins the race" comes from, right? And so really, when you think about chromatography, that's what it is, right? Some things are faster, some things are slower and lazy, some get hindered along the way by carrots or something, I don't know, and then some make it through. And so by basically judging and differentiating compounds based on how fast they move through your column or whatever, that's how you separate these things out, right? So we want to separate the tortoise from the hare, the slow and the fast. And we also want to be able to control when things flow through, right? Because say, for example, oh, I want the hare to only nap at this point, but then after a while, I want him to wake up and keep going, right? That's how we take advantage of these chromatographic techniques. So this is really where it all came from. People, they went outside, they took some leaves off trees, they mashed them up, and then they spotted that mashed up leaf mixture at the bottom of this chromatography paper. And then they dip this paper inside of some solvent. So that can be acetone or some kind of other solvent, right? And we've all seen this before, right? Why do paper towels work? Is because they absorb things. I guess more specifically, they adsorb things. And so as that, that front of the water is rising up because of capillary action, right? We've all seen how paper towels can absorb water into the paper crevices, right? The water, I shouldn't say water, the solvent, it can be acetone, whatever. The solvent brings the pigment up. And so as that level is rising, it's bringing up that pigment. And different pigments rise up at different rates. So in this case, the orange might be the tortoise who goes first, right? And then the yellow one might be some semi-tortoise, and this one might be a hare, another hare, right? So it's just like these things go up at different rates, and you can separate them out based on this sort of paper chromatography. Now, Leo asks, what's the difference between chlorophyll A and chlorophyll B? It's a good question. They're actually different molecules, and they have slight modifications. And so if you look at a plant, right, and look at their leaves, they're mostly green. And the reason why they're green is because of this pigment called chlorophyll. And it turns out that there just happens to be two different types of chlorophyll. Chlorophyll A, B, there's also C, actually. And that's what makes it green, and bluish as well. Yeah, there's multiple different kinds. It's pretty interesting. But it turns out that, when your leaves outside turn orange and yellow in the fall and they fall down and it's super beautiful outside, right? They have these other pigments too that we can separate out if we use this paper chromatography. And it's pretty cool. Now, what we're interested in, of course, in synthetic biology is proteins, right? We've heard so much about proteins these past few weeks during the summer, but really what we're trying to look at is paper chromatography. Yeah, Leo, carotene is definitely one of those. It's in carrots too. But what we're trying to do is we're trying to separate out proteins, right? Say you're growing some E. coli, some bacteria cells inside your flask, and you shake it around and it's growing nicely, and then you want to study a specific protein in that cell, right? You want to separate out that protein. Now, Colin and I have gone over a lot of things, right? So Colin's gone over SDS page, right? All the different blots, Western, Northern, Southern, all that stuff. Right? I've gone over isoelectric focusing. This is one more way, right? Protein chromatography has all these different subtypes, and we'll go over each of these throughout the remainder of the talk, and we'll see how we can apply the idea of tortoise and the hare in each one, right? Yeah, yeah. Oh, good question. What is RF value? So the RF value, what you do, this is not particularly important in terms of protein chromatography because we're not trying to sort of calculate each one, but we are trying to separate it. But what you do is you look at the line. For example, the solvent might rise up to this line, right? And then you also measure where your pigment is at. So for example, there, right? And then you measure that distance, you measure that distance, and then you measure that distance. And the RF value is just this distance divided by that distance, right? So for example, this one also. So this one actually isn't to scale, you can see, but yeah, it's basically the ratio of how far they travel compared to the solvent front. Yeah. Okay. Awesome. Now, the first one we're going to look at is this one right here. It's called ion exchange chromatography. And so really all these are, you can look at them from their names. So ion exchange chromatography, it's got something to do with ions, right? And so ions, as we know, are charged things, positive or negative. And so this is really hinging on the fact that proteins can be charged. Some are positive, some are negative. How do we separate them out based on charge? So I did do a talk on isoelectric focusing a few weeks ago, and that, I went over this a lot more in detail, but for now, just know that as you change the pH, the protein has certain chemical groups on top of it, right? It's got, NH3 or NOOH, and these things can have charges, positive or negative, depending on what pH you're at. And so, for example, in a really, really low pH, where it's really acidic, or you add a lot of acid, what you're doing is you're putting a whole lot of positive charge on there, right? And as you get more and more towards the higher pH values, you're putting more negative charge on there. So here's the idea. Proteins can be positively charged, negatively charged, or neutral, right? Now, how do we use this to separate out proteins via chromatography? Here's how, okay? And everyone feel free to ask questions in the chat because I really do appreciate you guys being engaged and asking questions. So here's the idea. You have some kind of column, and we'll see, it's the same theme over and over and over again. You'll have some column. You can see it in this case. There's this tube, and you fill the tube with a whole bunch of stuff, and that stuff just happens to be positively charged. So Leo asks, acid destroys stuff, but how do the positive stuff destroy things? You know what? If you see videos on YouTube of really acidic things, yeah, they blow up and they're really destructive. I agree. If you switch up piranha solution, oh my gosh, right? That burns everything. But yeah, so if you're too acidic, that's not good for your protein, right? If you're too basic, that's also not good. So there's a sweet spot. And so it turns out that ion exchange chromatography, it works well within maybe a range of 2 or 3 pH units. So you don't want to be too acidic or too basic. So you're exactly right. There's a sweet spot, a Goldilocks zone, right? Wow, all the fairy tales are just coming in today. The Goldilocks zone of pH, right? Yes, it is dissolved as well. Yeah, so these proteins are going to be dissolved in some solution, probably some kind of water buffer, right? And so these purple things are your solution. And the goal is you want to separate things out, right? You want to separate the positive and the negative proteins. You have some positive, you have some negative. Now, this column just happens to have a whole lot of these beads, and those beads are positively charged. So I mean, run wild in the chat. What's going to flow through? As you pour this liquid down, what proteins are going to be flowing through? Right? The beads are positively charged, and you pour your solution inside, and they drip down, right? What's going to be dripping down in the beginning? Yeah, so Nicole says positive molecules. You're exactly right. Wait, no, actually. So it turns out that, sorry, yes, my bad. I confused myself right there. Yes, you're exactly right. Positively charged molecules are going to flow through. Why? Because plus and minus attract. Exactly, right. If your column is packed full of these beads that are positively charged, then it's going to attract all the negative proteins, and the positive ones are going to be repelled. They're going to be like, get the hell away from me, and the positive ones are going to go down, right? So when you pour your solution inside in this positive bead sort of column, then your positive ones are going to be flowing through. Right? In this case, the positive ones are the, what is it, tortoise, right? The positive ones are the tortoises, and they're just going chugging along, right? They're flowing through drop by drop. But the Hare, they're like napping in the middle, right? The Hares are being trapped by the positively charged beads, and they're being slowed down because of this resin, because of this polymer that's positively charged. Now, this is just fancy terminology, but one of the molecules people use to form that positively charged column is called DEAE cellulose. So it's diethyl aminoethyl cellulose. You know, that isn't too important. What is important is that it's positive. So this is going to be attracting all those negative proteins. Now, my question to you guys is, well, all right, great. Now we have our positively charged proteins, and we can discard that, right? But how do we get our negatively charged proteins out? We want to purify it, right? We don't want it stuck in the column. How do we get it out? Feel free to answer in the chat. Remember, our negative proteins are stuck to the column. How do we get them out? We want to purify it, right? We want to separate it out. The slide kind of gives it away, but whoops. I should have made animation. Yeah, salt, exactly. But why though? Here's the question. Here's the important question, right? Why though? Why does salt make that protein want to go out? Right? The negative ones are bound to the positive, but then why does salt make that negative thing fall out of solution and into the thing at the bottom you're collecting it with?
I think I know why. Probably because all the salt there's lots of salt, so then probably for negative things are just like kind of pulled like this. And then there's another one below, and then it gets pulled and then pulled and pulled, and then it goes down probably. That's why.
Yeah. I think you're getting to the right idea. So Mavi says, Mahi, sorry, it splits into ions. You're exactly right. So salt, for instance, sodium chloride, the common table salt, right? It's Na plus Cl minus. And Andrew says more ions. Exactly. And here's a theme we're going to see over and over and over again today, right? It's the process called elution. And here's a fancy terminology word that just means, well, let's get the stuff we want out, right? That word, eluted. When we go through elution, we're changing something about the solution that you pour inside to allow the protein of interest to drift through. In this case, that's the negative ones. And so you guys are saying, oh, well, they're ions, and Leo says, well, they still attract. Yes, right? When you have a salt, you have a positive ion and a negative ion. And if that negative ion in your salt attracts to the cellulose more tightly, then it's going to say bye-bye to the negative proteins, right? Let me repeat that. If your salt, the anion, the negatively charged particle of the salt, binds to the cellulose more tightly than your protein of interest, then it's going to outcompete that protein, and your protein is going to flow through. Does that make sense? It's a competition, right? It's like, well, which one's more likely to be hindered, stuck by these positive resins? Well, it's probably going to be the salt, because the salt is a full negative charge, and the proteins are going to be eluted through. Now, I have a question for you guys, and you guys might have to think a little bit outside the box in this case, right? Because I didn't put this on the slide. High salt is one way to elute. What might be another way to elute this protein? Right? It's negatively charged right now. How do I elute it without changing the salt concentration? Let's say you're broke and you don't have salt for some reason. I don't know why. how else would you elute your protein? How else would you allow that protein to flow through? And I alluded to this earlier in the previous slide too. Andrew says react the DEAE with something. Yeah, perhaps. That's a good idea, right? So what if we change this to negative or neutral, right? That's a possibility, but it might be kind of hard. And also these things you can also try reusing them. So changing the column might not be the best idea. Leo says add more charges. I think you're getting close. Yeah, or ions maybe. This might give you a hint. Yeah, you guys a hint. Right, this slide. What can you change to elute your protein? Looking at this slide for inspiration. Exactly, right? pH. If we change our pH, then we can start eluting our protein. Why? Let's think about it in this way. Our protein is negatively charged right now because it's stuck into the positively charged beads, and that's why it's stuck, right? It's negative and positive, those attract. But what if we can change the pH such that we can change the charge on our protein? If we make the pH lower, we make it more acidic, that'll what? That'll make the protein more positive. And then what? Then it'll flow through because then the positive and positive repel. Right? So by making it more acidic, we can also elute. So there's two options. We can elute with high salt or by changing the pH. Right? And this specific ion exchange chromatography is called anion exchange chromatography because a protein of interest in its native form is an anion, it's negative. So we call this an anion exchange column. Right? By making it more acidic. Yeah, exactly. So pH allows us to elute more. Perfect. So here we see the example in the previous slide, that was anion exchange because the column was positive, the proteins were negative, and they attract. That gets stuck, right? There's also a case where some proteins are positive, right? Then you would use a cation exchange column. Same idea, but it's just flipped. The column is negative and the cation protein is attracted, right? And of course, same idea here. We can also elute here with charge by changing the charge by making it more basic, the opposite, right? If we increase the pH, then this will become neutral or negative, and then it will elute. Same idea, right? But opposite. Cation and anion exchange chromatography are different types of ion exchange chromatographies. Okay, and here's one more diagram to get the idea through, right? Now, here's another tricky point, right? When we're thinking about chromatography, there's always the idea of, well, some are negative, some are even more negative, some are even more negative, right? And so there's like a gradient, and this is like really annoying as well because as a synthetic biologist, you really want your thing only, right? Let's say your protein was, I don't know, -2 charge, and there just happened to be a protein that was -1 and -3, right? How do we control exactly when our protein elutes through? Honestly, a lot of trial and error. So you try it, and I mean, Colin's laughing, but it's a tough, yeah, it's a tough game you have to play. But a lot of protocols online, they say, oh, add this much salt, or oh, change the pH by this much. But really, if you're studying a new novel protein, you have to trial and error, right? So it's tough. And so you can add a little bit of salt, add more salt, and add more salt, and then your proteins can elute depending on their charges and how much charge they have, right? Okay, let's move on. What time is it? Oh gosh, I need to stay on time. Okay, 7:21. Next up, gel filtration, also called size exclusion chromatography, SEC, right? SEC, size exclusion chromatography. Now, you guys, this is going to become second nature to you because because we just went over the ion exchange chromatography, right? So by now you can already begin to think about, well, I used ions. I took advantage of the fact that proteins form ions to separate. I mean, can you guys guess what this is taking advantage of? Well, obviously this can be size, right? So here's the idea. Different proteins are different sizes, right? There's a whole variety of, protein shapes and how they fold. That's a whole field of study, right? But the basic premise is, oh, well, some proteins are bigger, some are smaller, right? Check this out. ATP synthase, massive protein, right? Whereas insulin is a bit smaller, right? If you want to study, for example, I don't know, titin, probably the biggest protein there is in your muscles, right? Super big, right? So there's all these different protein sizes. And so the idea is, well, what if I can separate them out based on size? Now, of course, Colin already talked about SGS page. That's one way, right? But the problem with SGS page is you can't really purify. It's more for analytical purposes, right? You can see it with SGS page, but you can't really purify. You can't really get a lot of it out. It's only with that one gel that one gel sort of cartridge. But when we want to purify something, we really want a lot of it, right? We want as much as we can. We want to purify a good percent yield from our cell culture. And so we don't want just that one lane in the SCS page. We want a whole lot. And that's where SCC comes in. That's where size exclusion chromatography comes in. Some are big, some are small. Let's take advantage of that, right? So here's the idea. Yeah, oh wow, yeah. Insulin, trypsin, hemoglobin. Yeah, so proteins have a huge variety in shape and size. And so here's a basic premise. Okay. Titan, yeah. And so some are small, like the yellow one, teensy weensy, right? Some are medium, orange, right? Medium in size. And some are huge. This red protein is one of the bigger ones, right? You can see it's got 3 big blobs. And so the premise is, well, let's change our column to match our needs. What is our need? We want our column to be specific to certain sizes. Okay? Now, imagine that the red big blob is the tortoise, right? And the tortoise, slow and steady, wins the race. He just keeps on chugging through, right? And that tortoise has some perseverance. Wow. Like, I wish I had that perseverance. And so the tortoise is just chugging through to the finish line. That's these big proteins. Right? These big proteins, you pour it in your solution. It has these beads with all these pores and these crevices, right? And you pour your solution through. And the big proteins are like the tortoise. They're like, what the heck? I want to just go through, right? Just let me through. I want to chug along and just go to the finish line. That's the big one, right? But the medium one's like, it's kind of like a hare. And the hare, what did the hare do again? The hare was, taking a nap on the side, getting distracted, finding carrots along the way. Not sure if carrots are actually in the story, but regardless, right? The hare was distracted, right? The hare got sidetracked with all these different things. The orange one's like that, right? The orange one's like, hey, look at this. Let me explore this crevice. Hey, look at this. Let me explore here. Let me explore here, right? So what's happening is the orange one, let me do orange color, right? The orange one might go here to explore, it might go here to explore, it might go here to explore, and then go through, right? So the orange one was like the hare in that it was sidetracked and hindered and slowed down by his nap time or his adventures, right? The yellow one, even more so, right? So for the yellow ones, let me draw this in orange again, right? The yellow ones, the small ones, are even more adventurous. They're even more sort of sidetracked. And so they can go in here and here and here and here and here and here, and then they go on through, right? So the idea is that the small ones are trapped in these crevices and then they go on through, whereas the big red one, oh gosh, these are like the tortoise just chugging on along. So what does that mean? That means that the red one follows a shorter path and the smaller ones have a more convoluted path. It's longer, right? So how does it separate it by? Time. The ones that elute first are bigger because they have a shorter path to follow. The ones that elute second or third or fourth or last, those are smaller. You can separate proteins out by size. That's the idea of size exclusion chromatography, right? So why does it do that then in real life? Yeah. So in real life, it physically goes into these crevices inside of these pores, right? So that's how it works. Yeah? Okay. Any more questions? Please ask them in the chat. Okay. And here's one more image explaining the same thing. Now, we have all these beads with all these crevices and pores, and the small ones get sidetracked. They go inside these pores. And the big ones are like, nah, I ain't got time for that. And they just go straight on through, right? So you can see how the big ones, they can't enter the gel until they elute sooner. And the small ones, they go through this convoluted path and they elute later. So you can see, so this might be a new term here as well. It's called a chromatogram. It's essentially, how do I explain this? It's a plot where you have some dimension on the x-axis, in this case it's time, and you're measuring the protein on the y-axis. So there's many ways you can measure proteins. I won't get too much into details, but there's ways you can use UV light to measure how much protein there is. So you might have like a UV detector right here in the bottom, shining UV light, right? And then there's some detector here. And so as your protein goes through, the UV light will be absorbed by the protein, and your detector will be like, oh, there's a protein there. So you can see when the protein goes through. And then you can see how, oh, this red one gave a signal first, and then the pink one gave a signal. Ah, it was SEC. Therefore, the red one, this red curve, it represents a larger protein. This pink one represents a smaller protein, right? So any questions about this? Is this like how electrophoresis works by size? Yeah, same idea, right? Well, actually, not really. I mean, Colin, you can correct me if I'm saying this wrong, but in terms of the size exclusion chromatography, the idea is that these small particles can go into these crevices. In SDS page or in electrophoresis, the smaller ones can flow through faster. So it's almost the opposite.
Yeah,
Yeah.
But similar idea.
Right. And so I think in terms of electrophoresis and SDS page, the reason why the small ones go faster is because the pores themselves are different. And so because if you have these pores, the smaller ones can navigate the pores better and travel faster. But in this case, because of the way that the beads are designed, the small ones actually get stuck instead of going through faster. And so it's the way that the gel's made. It's kind of cool how they actually reverse the way that these things work. But yeah, good question, Mavi. Okay, let's move on. And here's your one-way diagram, right? You have some big ones, some small ones, and you elute the big ones faster and the small ones later because of this size exclusion chromatography. Good question. And to that, I point you towards our YouTube channel. I'm kidding. So electrophoresis is the idea that you apply some charges, and then because of the electric current that's flowing through, that can carry the proteins through, and depending on the protein size, you can make them travel through different rates. And so I won't go too much into detail here, but electrophoresis is kind of similar in where you're trying to separate out some stuff. It can be used for DNA or protein or RNA, but electrophoresis is essentially the same idea, but in smaller quantities. So it's not used for purifying, it's used for analytical techniques. You can see if it's there or not. So, thank you, Colin. Sweet. Okay, let's move on, shall we? What time is it? Half an hour left. Okay. So the next idea is hydrophobic interaction chromatography. What does hydrophobic mean? All right. Someone answer in the chat. What does hydrophobic mean? Yeah, water hating, right? Hydro, water. Phobic, hating, right? Just like, I don't know, claustrophobic or, arachnophobic. If things are hydrophobic, they hate water, right? Of course, I hope that you're not hydrophobic because you need to be hydrated, right? But proteins can be hydrophobic. And so what that means is that there are some amino acids that don't like water. I won't get too much into the details of the biochemistry here, but proteins are a change of amino acids, and those amino acids have different properties. Just like they have different sizes, they have different charges, they can also have different polarities. So some amino acids are polar. They like water. The charged ones also, they like water, right? So they are hydrophilic. There are also other ones that are hydrophobic, like phenylalanine or alanine, right? I have a shirt from high school, and so in high school we had sort of like these nicknames that we had to make on these shirts, and mine was alanine, and so yes, yeah. So alanine is hydrophobic, exactly, and so, and also fun fact, phenylalanine, it's because it's alanine with a phenyl group on top of it, right? That's also hydrophobic, and so there are different hydrophobic amino acids, and those hydrophobic amino acids, because of their properties, they can make the protein behave in a certain way. And here's how. For those of you who might have taken a bio course, you'll know this already. But just as a quick review, you have some hydrophilic amino acids, things like serine or tyrosine or histidine or lysine. Those water-loving amino acids are in blue, and water-hating hydrophobic are in red. And so I feel like commonly we think about proteins and we just think about the amino acids themselves. We think about them in isolation. But in reality, what's happening? There's a bunch of water, right? There's solvent around it. And the water, it actually impacts the folding a lot. And so Colin's going to do a protein folding, a webinar all about protein folding next week. And so for now, the basic premise is proteins fold into a shape depending on their amino acid sequence, right? So for example, you might have methionine, alanine, phenylalanine, histidine, and serine, right? This order of amino acids then determines the protein's final shape. And you would expect the hydrophobic ones to be on the inside. It makes sense because the water's on the outside, right? The solvent's all here. I can draw the blue water waves around here, right? Water's all around. And so if you put water all around here, right, this protein's mad, right? Why? Because this one is in the presence of water, right? These are all seeing water, so these hydrophobic ones are all really sad. But how do they become happy? They're scared of water, right? They want to get away from water, so they want to be on the inside, right? Oh, no, sorry. Now they're happy on the inside. Sorry. So they essentially fold inside, and it's actually a huge problem. This idea of protein folding, and I think Colin will get into that more next week. But how do we predict the structure from the amino acid sequence? That's a huge topic. It can solve a whole lot of problems in terms of pharmaceuticals and protein structure determination and a whole lot of things. So now back to the question again, right? How do we separate? How do we separate proteins based on their hydrophobicity? Well, how many hydrophobic amino acids there are? Well, I mean, the obvious solution is to have a column that's also hydrophobic, right? That should make sense to you guys, because think about it this way. If you have some protein, right? Of course, initially the protein won't have its hydrophobic region on the outside, it'll have it on the inside. But say you also have some hydrophobic column, right? Pretend this is the column here, right? And pretend that this hydrophobic column has hydrophobic things attached to it. I'm not exactly sure what the chemical is, but there's some hydrophobic group attached on the walls of that column or in the beads of that column. And so as your protein is flowing through, then the hydrophobic parts of the protein and the hydrophobic column are going to want to interact, right? Right now, this is sad and this is sad, but if they're interacting like that, they can minimize their surface area to water, and that's how they can bind to each other, right? It's this hydrophobic effect that's going to bring them together. Now, Leo asks, what happens if it's all hydrophobic? That's a great question. You know why? Because rarely will you find a protein with absolutely no hydrophobic amino acids, right? So there's always going to be, maybe 1 or 2 or 3 or 10 or 100 or 1,000 amino acids that are hydrophobic. And that's the key idea. I think, Leo, you're getting to it, right? Because some are more hydrophobic than others, right? I think you're thinking along the right way, because, what about, if you have some that are very hydrophobic and some that are less hydrophobic, well, how do you separate those? Because they're all going to bind, right? So Leo asks, it'll be spinning crazy, probably. This will probably disrupt folding, right? So the protein might fold a little bit and it'll maybe spin a little bit to interact these two together, right? So the folding will change probably. Proteins bind because of polarity. Yes, I would say it's almost the opposite, but I think you're on the right idea. They're binding because of non-polarity, right? Because they're both non-polar, they want to minimize their surface area to water, so they want to attract each other. So I think it's kind of like a indirect effect, right? It's like the water wants to be associated with each other, not to the hydrophobic parts. So it's almost like the non-polar regions are attracted to each other indirectly. It's not that they're attracted to each other, because they have some inherent attractive force. It's because of the solvent, right? If it was all hydrophobic, wouldn't the protein not fold at all, since there's no water to get away from? Well, I would say there's still water, because water is a solvent, right? I would say it will fold in a certain way. And it will fold in a certain way such that the hydrophobic interactions are minimized with the water. And we'll see examples where some proteins are more hydrophobic and some are less hydrophobic. But the question still remains, right? How do we separate them? Because if they all have some hydrophobic component, they're all going to be interacting with the column, right? How do we manage to separate these based on their hydrophobicity, right? So again, we see the same process, right? We first bind it, and then we wash away the weakly binding ones, and then we elute, right? So we see the same thing over and over again. It's the same theme, right? Chromatography, protein chromatography, it's the same theme. We first put our solution through, then we wash it to get rid of all the weakly binding ones, and then we elute. Now here's a big question, right? How do we specifically elute the ones that we want? What do we use to elute? And here's the answer. Salt. Again, right? Why? Why does salt help us elute this from this protein column, from this hydrophobic exchange column, right? Any guesses? Right? Think about the hydrophobic interactions and think about how that might change depending on the salt concentration. And this might give you a hint, right? This diagram here. As you lower the salt concentration, the proteins are going to want to elute. But if you have really, really high salt concentrations, then the proteins are going to be more bound to the stationary phase, which is the column. Any guesses as to why this is the case? Why there is this trend of low salt means elute in terms of hydrophobicity? Magnetic. Good idea. I don't think so, but I love that thinking though, right? There are actually cases where you can use magnetic beads to isolate and purify proteins. That is a thing, but not in this case, no. Right? Salt, think about, so for example, sodium chloride, right? There's some positive, some negative things. How would that impact the folding of the protein? And before I move on, let me just clarify some terminology. I meant to talk about this earlier, but I guess I forgot. So before I, confuse everyone, the stationary phase, it's stationary, it doesn't move, right? So for instance, the beads that we saw in the ion exchange chromatography, the beads with the pores in the size exclusion chromatography, and the column in this case with the hydrophobic things, that's the stationary phase, right? They're still. The mobile phase is the one that's moving. So our sample is the mobile phase. And so we want our thing to attach to the stationary phase and then elute after we lower the salt concentration. Leo asks, do hydrophobic things hate oil? No, they actually love oil. Because hydrophobic things are water hating. Guess what? Oil hates water too. They're both hydrophobic, so they love each other. So hydrophobic things
that
actually.
means hydrophilic things hate oil.
Hydrophobic things actually love oil. They hate water. They both hate water.
Hydrophilic things.
What's the difference between hydrophobic and hydrophilic?
Go for it, Colin.
All right. Yeah, philic means love. So like hydrophilic things will love those negative charges. They'll love water and things of that sort. Oil has no charge. So hydrophobic things will dislike things with charges, but love things with no charge, like oil or
Right.
things like that. It's a way to think about it too.
Yeah, thank you, Colin. So Leo, have you ever poured water and oil together? Right? What do? They separate. Why? Because oil hates water. Right? So same thing in this case. There are some proteins that have hydrophilic regions. And so here's the answer. All right. It turns out that in high salt concentrations, think about what the protein would rather interact with, right? If you have a whole lot of salt, the water would rather prefer to interact with this huge amount of salt than the protein. So that means that the protein will rather interact with the hydrophilic beads on the stationary phase. Does that make sense? Right? If I have a whole lot of salt, there's all these positives and negatives. Water loves that, right? It's hydrophilic. And so water loves all the salty things. Not saying water's salty, but I mean water loves salty things. And then if you have a high salt concentration, water will bind to the salt rather than the protein. So basically the protein's more lax. And the protein can then expose its hydrophilic regions. Oh, sorry, it'll expose its hydrophobic regions to interact with the stationary phase. Right. So Leo asks, what's the difference between hydrophilic and hydrophobic? Yeah, so hydro means water, and philic means loving. So hydrophilic means water loving. It loves to be with water. So for example, salt is hydrophilic. Hydrophobic means water hating. So there's a whole lot of amino acids that are water hating. They're hydrophobic, right? And so as we change the salt concentration, then we can start eluting our proteins. As we lower the salt concentration, we can pour maybe, I don't know, 10 millimolar and then 5 millimolar and then 1 millimolar of some certain salt. Then we can start eluting our proteins by this hydrophobic exchange chromatography technique. Yeah? Any questions about this? Okay, and here's an example of proteins that have differing hydrophobicities, right? So in this case, cytochrome C has very little hydrophobicity. And so that means that at very high salt concentrations, it will elute because it'll prefer to associate with the water and the salt and go through with it, right? And things like chymotrypsin, it's an enzyme, right? It has a lot of hydrophobicity. And so that means that it'll elute at pretty low salt concentrations. And so you can see there's this UV absorbance that's measuring when these proteins elute from the column, right? And so in this case, I guess the chymotrypsin would be the Hare, right? It's getting sidetracked by all these hydrophobic interactions, and the cytochrome C is the tortoise, right? Let me get on through. So at high salt concentrations, that's when it'll go through. So again, salt's really important when we're thinking about these elution techniques. Okay, let's go to the last one before I open it up for questions. So affinity chromatography, this has got to be my favorite one because this really relies on, looking what nature has and taking advantage of what nature gave us. So let me tell you what I mean. Here's an example, a scheme of the generalized affinity chromatography, right? What does affinity mean? If you have an affinity for something, you really like it, and you want to bind to it. And so if you have an affinity for, I don't know, cake, you love cake, right? You want to get to cake, you want to get your hands on that cake. If you're a protein, you might have certain affinities. And so that affinity has to do with receptors. And so for instance, if you have a column, again, we've seen the same thing, right? We first pour a sample in, then we wash it, then we elute. And so it turns out that if our column has a receptor and our protein has an affinity for that receptor, there is this affinity, there's this binding event that takes place that prevents these proteins from going out, right? I have an affinity for books. Nice, nice, nice. Yeah, we all have our hobbies, our affinities, right? Proteins do too. I mean, proteins are people too. No, that's false. But proteins have affinities too. And so, for example, this hexagon fits perfectly inside of that hexagon-shaped receptor. So that means that as you run this column through, then the proteins that don't fit are the ones that are washed out. And you're only left with the ones that fit into the receptor site, right? And so if you think about this case, and I'll give some more specific examples later on as well. I want you guys to think about how do we elute, right? And this draws back on the concept we learned about in the previous ones as well. We have this binding event. How do we elute? What do you guys think? This hexagon is binding right in there, right? So how do we elute this protein? How do we get it out? They don't show it here, but there's something we add to make it elute. And this draws on a previous concept we learned as well. Leo guesses you change the hydrophobicity. That's a good idea, yeah. So if you were to somehow able to change the conformation, right, that might allow it to escape, but there's actually an even more big brain way to do it. Or pH, maybe, yeah. So pH might also change the conformation to make it binding more or less, Andrew says salt, perhaps. Even more big brain though, right? So think back to, these are all great ideas, right? So I'll give you credit for these because I think these probably will work, but there's something that works even better because if you add salt or pH or if you change the conformation, you might mess up your protein. And so if we want to purify these things, you typically want to keep the pH constant. It is an option, of course, but if you want to keep it in its native shape, you typically want to keep that stable. Right? You don't want to change pH. You don't want to. It's an option, but you want to avoid it for the most part. Right? That's the whole point of buffers. You don't want to change the pH. What might we add to allow this hexagon to elute? And this diagram is kind of misleading. I'll give you guys that. Think back to our discussion about the ion exchange chromatography, right? What did we do for the ion exchange chromatography? Well, we added some salt, right? And that really allowed some charges to compete with the other proteins, and that allowed the protein to elute, right? Depending on the, yes. So Andrew says an even stronger binding protein, right? That's the sort of optimal way, right? Think about it in this case. We have our hexagon binding in these sites, but Andrew says put an even stronger binding protein in there. Put a hexagon-er thing in there, right? In this case. So if you put an even more hexagon-shaped thing somehow, then that will competitively bind to this thing and it will allow our protein of interest to elute, right? So here's the concept. We want our protein to bind, but we also want something even stronger to bind even stronger to it, to compete for it, to allow our protein to elute. Right? So that's the idea. We want an even hexagon-er shape. And so in this case, our protein of interest is the hare, right? Being sidetracked, lazy, taking naps along the way, right? It's taking a nap in this bed right here, this receptor bed. And the hare loses the race, but hey, that's okay. It's what we want, right? We want to separate these things. Whereas these green, yellow, and purple ones, they're just booking it, right? They're the tortoise. And so let's look at one specific example, right? And this is an amino acid called histidine. we see there's this backbone, this chain, and they have these histidines coming out of it, right? If any of you attended our previous webinars as well, we had one talking about pymol and excavator fraction. This is how we purified it. We used this histidine way to purify our protein to crystallize, hopefully, next year. And so here's the idea, right? DNA is super cheap. We can use our DNA to encode a certain set of amino acids, right? And so, say for instance, at the end of our protein, we add the DNA to allow us to code for a whole bunch of histidines, right? Also, Andrew asks, what are the benefits of chromatography versus SDS page? Very good question, right? So they're both separation techniques. SDS page, and Colin, you can add on as well. I would say SDS page is more for checking it's there. It's analytical, whereas chromatography is for actual purification and getting a batch full of this thing. So
That's exactly right. Yeah.
yeah. So there are some differences, but they are both relying on the separation technique. And so here's the idea. You have some column, you have some beads, and the beads happen to have this compound here. They have some nickel compounds, and they have this triacetic acid compound, right? It's called nickel NTA. Oh, NTA stood for nitrilo triacetic acid, something like that. And so you have this nickel right here, right? Nickel's positively charged. So it likes what? It likes electrons. It likes negative things. Guess what? These histidines are chock full of those, right? These histidines have a whole bunch of electron pairs, and so it will bind pretty tightly to this nickel. That's what we use to separate this out, right? By having this hexahist tag at the end of our protein, we can then essentially run our protein through this nickel NTA column, and then all the proteins that don't have this histidine chain will then pass through, and then we can elute our protein of interest, right? And so here's the idea. We have our protein binding to nickel, right? Good. It's the Hare, it's lazy, it's binding, taking a nap on the nickel at the column. How do we elute? What do you guys think? What compound should we elute? Should we use to elute this polyhistidine tagged protein? So Andrew said it before, you want to use something that binds even tighter to this thing, right? What might be that thing? Even more positively charged protein? I wouldn't say positively charged protein because the positive and positive will repel, right? If anything, you probably would want something negative or some kind of electron-rich thing, right? Why not match it? Let's use imidazole. There you go, right? So imidazole is like histidine, but only the R group, right? So here's our histidine amino acid. You see in the previous slide, this is a whole bunch of histidines, right? So a bunch of these. Let's just pour in a whole bunch of imidazole in there, right? 500 millimolar or 1,000 millimolar, right? So by pouring, I guess that's just 1 molar. So by pouring a whole bunch of these imidazole molecules inside that column, you are outcompeting this protein with a polyhistidine tag. And if you outcompete it, what happens? It elutes. Right? So therefore, that's how you would get rid of your protein. And so really, it's troubleshooting, again and again and again. When we were trying to elute our DMJ Hampty protein, we must have run like a whole bunch of columns out because we were like, well, this didn't work. It kept getting stuck. It's really annoying. Like if you don't really lyse your cells well and you don't filter it off really well, the cell pieces get stuck in the column and it really jams it up. So, trial and error is really important. It's really annoying, but you gotta go through it. So that's what you do. You make your protein with the polyhistidine tag, and then you use imidazole to elute it from your column. Okay, very good. Now, typically, you'll also add some kind of second least favorite experiment. Yeah, true. You'll typically also want some kind of cleavage as well. So you want to have some kind of amino acid sequence that can be cleaved off by some kind of enzyme. And so then you'll get rid of this tag afterwards. So it's tough, it's hard work, but it is useful at the very end, right? One more example, GST, right? This stands for glutathione S-transferase. This is one more example of affinity chromatography. So this GST tag, it has an affinity for glutathione. I mean, it makes sense, right? It's an enzyme that binds to glutathione. So this GST binds to glutathione via that bond, it has an affinity for glutathione, right? So this is the Hare. It gets stuck, right? It's lazy. It's being stuck on the column, and we can't get it out unless we use glutathione, right? Then we can competitively bind to this GST protein, and then we can elute it off the column. So you guys are seeing the same concepts applied over and over and over again, right? And I hope you can apply this to other things as well. I have this protein of interest. Let me calculate its properties and determine what's the best way to purify it or to analyze it, right? And so here's one way. You can attach this GST enzyme to the end of it and then elute. And at the very end, you'll probably also want to cleave there to get your native protein, right? So very good. Okay, and etc. Right? This is really 4 of the, I want to say, the most common ones. Of course, there's a whole bunch more, right? So really, I think these 4 illustrate the concept pretty well. You want to run your sample through it, it binds somehow, and then you wash it to get rid of the weakly binding ones, and then you elute using some specific buffer, right? You elute it with glutathione, or you elute it with imidazole, or you elute it with salt or no salt, right? So the idea of bind, wash, elute, that 3-step concept is applied over and over and over again in all these protein chromatography techniques. Tortoise and the Hare, right? It's the same story said over and over again. And of course, why stop there, right? There's a whole bunch more. Like, oh my gosh, there's so much. But there's so much more that you can do with chromatography, right? So paper chromatography is what we talked about in the very beginning, where you just dip it inside of the water or acetone, some solvent, and it rises up in the paper, right? That's paper chromatography. There's also thin layer chromatography. If you ever take orgo, you'll do that. So TLC plates, you'll do that a lot. Colin shaking his head. Yeah, I feel you, man. I didn't really talk a lot about phases because in all the ones that we talked about, we went with a solid stationary phase, the beads, right? And a liquid mobile phase that flowed through. But you can also play around with the phases as well. And so for instance, if you had gas chromatography, you actually want to, what's the word? Not boil, but take it into the gas phase. Not boil, but Colin, what's the word? Do you know the word I'm talking about?
No.
Like,
Sorry.
like take it from the liquid phase, vaporize? Yeah, vaporize, sure. Vaporize, yeah. You want to vaporize your protein, and by vaporizing your protein, you turn it into a gas, and then it can flow through another column, a gas chromatography column, and it will travel along this liquid stationary phase, and it will separate out like that. It's another way to separate things out. So you're not limited to just liquid, right? You can also vaporize your protein. Of course, by doing that way, you probably can't get it back. You probably could, but it'd be hard to, I think. So I think by sticking with the liquid phase, you can probably, you'll be easier off, sticking with the liquid phase. But this is a thing. So typically, gas chromatography is also used for analytical techniques. So for instance, if it doesn't work in the liquid, for instance, it's not soluble or something, there's some problem with liquid, you can vaporize it, it's always an option, and then analyze a protein. You can see, it is a separation technique, and you can see, oh, is this protein present or not? So all of these can be used for analysis as well. There's also, HPLC or FPLC, and we saw these, right? We saw size, ion, we saw all these things. So there's all these different options, and you want to choose the best one for your experiment, right? Because I think it's good to look at the variety, because then you know what to choose from. Of course, you can have too much of a good thing. You're like, oh my gosh, what do I choose? There's so many options, right? I mean, again, trial and error, right? Read literature, read papers, see if people have done it before, right? See what worked for them, see what didn't work for them. So I think a lot of it's experience and trial and error. So choosing the right chromatography technique to go through is definitely a thing you just have to guess and check. But yeah. And here's one more example of a chromatogram, right? So you can see, oh, we apply our sample, something goes up. This is what we don't want. Right? Then we elute the thing we do want, which is protein B. And you can see that there are these peaks which represent when that protein came out. And so, yep, then you celebrate because you got your protein. Yay. So are there any last minute questions? I know that was a whole lot of techniques I just went through. Chromatography is not one thing, it's a whole bunch of different techniques. But are there any questions at the very end?
I got a question for you, Alan. So in your chromatogram, you use time quite often. Why is time important? What can you use that for?
Why is time important? So I mean, especially in things like size exclusion chromatography, where some things are stuck in crevices and some pass through faster, some are slower. Depending on you know what time your protein elutes through, that's how you would separate out your protein. You know, Oftentimes we use time because we can say, oh, we pour in this elution buffer at this time and then elute at that time. But typically, these chromatograms, it's kind of tough, especially if you're doing it manually. But if you had it in a machine, so for instance, there are HPLC machines and there are FPLC machines. If you have a machine that can automatically record it for you, that'll be easier to do it via time. So, all right.
Thank
Is
you
it
so
going
much.
good? Yeah, it's going good. Yep, it's going fine and dandy. Yeah. So, I mean, why stop there, right? I mean, we have these techniques. Why not combine it with other things? So I'll use these last 2 minutes to talk about what's called tandem chromatography. What does tandem mean? You guys seen tandem bikes, right? I've never ridden one. I've always wanted to. I mean, take me out for a ride, Colin, later, but sometime next year. But these tandem bikes, right? You have two people on the bike and they're pedaling together and they go together, right? There's also tandem chromatography. So what is tandem chromatography? Yeah, let's do it. So tandem chromatography is, well, let's run it through the chromatography column or whatever it is, and then let's put it into something else. So what if we had a liquid chromatography machine connected to a mass spectrometer or an infrared spectrometer machine or some NMR machine, right? By having this tandem chromatography techniques, you can then run your protein through, separate it, and then analyze it immediately, right? So LCMS, liquid chromatography, mass spectrometry, right? You can start combining the techniques you use to, separate and analyze in tandem. So, all these different good techniques that you can do. Okay, I think we are pretty much out of time here, so I thank you all for coming, and I'll hopefully see you next week when Colin comes with his fascinating Rosetta webinar.
Yeah, yeah, we're doing Rosetta Commons next week, which is a way to simulate protein folding. We attempted it last week and ran into some problems, so we're going
Yeah,
again.
yeah. All right, I'll stop the recording and